Hi, everyone. I'm Jeff Petruska from the University of Louisville, where we have the home of global hub of UPS, really great horse racing, and the world's best bourbon. I need to tell you guys that I have some disclosures here associated with a couple of companies, but nothing I talk about today is going to have anything to do with that. They have nothing to do with this. I want to thank the organizers for putting on this meeting. It was one of the best meetings I've ever attended in the past, and I really enjoy it. I think it's great for the community, and it's nice to be here among these other panelists. So thank you. I'm going to talk today about some things impacting clinical trials. Everybody here knows that clinical trials are difficult enough. It's hard to pull enough or recruit enough people from such a small group, and all of the folks have very different sorts of injuries, even if some of the classifications lump them all together. And that's just the start of it. That's just the design. Clinical trial success is further endangered by noisy results, things that don't always work. You can have people who are responsive to your treatment. You can have people who are unresponsive to treatment, and it's unexpected. By all accounts, they should be reacting the same, but they don't. Biomarkers can be used to clarify the inclusion and exclusion criteria for the design, and can be used for post-hoc data parsing. So one of the things I just want to say is that I'm introducing the biomarkers as a general concept for sort of the framework for the talk, and I'm going to get into some details now. One of the things that we've been working on, myself and some colleagues, both at U of L and elsewhere, is all about the circuitry that's local to the below-level spinal cord. A lot of circuitry exists in the spinal cord below an injury, and it still functions. Doesn't always function the way it did before the injury, of course, but it can still function. An example is reflexes. This is a tendon tap reflex. You do not need a brain for this, which is good. And it's necessary. A lot of even more complicated functions include circuitry that exists below the spinal cord injury. So those things are still working, not the same as before the injury, but they are still working after an injury. And that may be good because it gives therapies something to latch onto, something to use, but it could end up being bad too. One of the things that my colleague David Magnuson has started doing at the theme of his laboratory is asking questions in the laboratory, basically treating rats like humans, and asking questions of what we're doing, is it good? And one of the things that they started looking at was therapeutic stretch. They worked with physical therapists to design stretching paradigms for rats that would mimic the physical therapy that a lot of spinal cord injured folks get. Even normalizing the forces and the range of motions, et cetera, is really nicely done. What we're looking at here is a recovery over time of the BBB score, which is basically locomotion for rats, and it recovers to a certain point after the injury that they've all received, and it's going to stay that way. We know very well, the field knows very well, it's not really going to improve unless you do some pretty significant treatments. Well, one of the things that David did was to stretch the animals, and lo and behold, when you give stretch, you get a very rapid, very strong, and very long-lasting suppression of the locomotor ability that the rats retained after their injury. It was very surprising and very disturbing, and we started asking a lot of questions about this. Now, one of the things David said was, hey, Jeff, what do you think might be going on here? We went over some possibilities, and one of the things that I said was, David, we know that the nociceptors, the pain-sensing neurons, have a lot of influence over the motor programs. This is exemplified here, and we've all probably experienced. You can be walking along, stub your toe, step on something sharp, and generally you don't fall over. And that all happens before you even realize that you've been hurt. The circuitry that is protective and extends your other leg and picks up the leg that's being hurt, that's all spinal cord. And so the local reflex circuitry is still functioning below the injury. And importantly, the nociceptive reflex circuitry actually has override authority for the motor programs. It can stop them from going, even though your brain sent down, hey, you should be walking. Nociceptors say, stop. So we did an experiment where we took out the nociceptors in some rats. And it turns out that if you stretch those rats without nociceptors, you don't get this suppression. What that tells us is that a locomotor suppression by stretch requires nociceptors, requires those pain receptors. And so that's going to play into a lot of what I'm talking about here. Activity from the nociceptors may be influencing a lot of what we see in the lab, and a lot of what folks see in the clinic and in human research. Of course, that's one of the big questions. Great, we see this in rats. It's telling us something. It points us in a direction, but does this actually happen in humans? So David Magnuson has been working with a few of the clinical researchers around the country, some of whom are here on this panel. These are some bits of data from the Emily Fox lab down at the University of Florida. And I'm going to give some examples here of a small group of folks that they, with whom they worked, to ask the question, when you do stretch, does it influence their function? Is it doing bad things the way it did bad things in the rat? And the answer here, at least for this individual, is no, not really. The stretching, which is on the right side of all of these, so muscle strength here, quadriceps spasticity here, walking function here, and this is after a sham or after a stretch session, pre and post. Not much change. This is not very striking. So looks like it's fine. But then you go to somebody else. And a walking function, this is a very common test that's used across the country, across the world. Again, it looks like stretch really doesn't hurt anything, according to this test. Why don't you go look at some other tests that are a little bit more, maybe labor intensive, and a little bit more focused. Different story. Strength decreased, spasticity increased after stretch. So here we have, two examples. One where stretch didn't do anything, one where stretch did something, but not in all of the functions. And then I thought it was very striking in their data that when they asked the subjects for their own ratings, what's the perception about the effects of stretching on these characteristics that you normally have? Perception of a negative outcome only shows up when you do stretching. So that was very interesting. And again, small group, but it's kind of telling. So I want to be very clear. This is not a slam on the practice of stretching. And this can be spoken to much more in depth by some of the folks on this panel. I've spoken to some of them about this. Stretch can definitely be very helpful for some people, clearly, as even some of Emily's data showed. We can continue to learn about therapeutic stretch, and we will continue to learn about it. And the stretch that I'm presenting here is as an example of the concepts that there can be things that we do or things that are happening that can interfere with outcome measures that we use in clinical trials, and that the folks living with spinal cord injury have to deal with every day. So one of the directions that my colleagues and I are headed is looking at those nociceptors. Why are they such a problem after spinal cord injury? And one of the things that happens is that their effects are unchecked by the usual top-down control. There are lots of things that happen before spinal cord injury in a spinal intact situation where you might have activity of nociceptors, and that activity goes into the spinal cord. It would do something, but the brain says, yeah, no, I don't want to pay that much attention to it. And that's a normal system, but that might be gone. That control and modulation may be gone after spinal cord injury. They become easier to activate, and they have a greater effect after spinal cord injury. And that really brought up some interesting thoughts on the scientific side. How does that happen? How do the nociceptors, the pain receptors, our sensory neurons, how do they become more active? Well, one of the things is that their cell bodies are sitting in the same cerebrospinal fluid as the spinal cord. And if there's been an injury, maybe there's something in the CSF that is making those sensory neurons more easily activated and more responsive. So we did an experiment. We got some cerebrospinal fluid from the Brian Kwan lab, from a Yucatan mini pig that had been injured a month earlier, and put that CSF onto some sensory neurons from rats that did not have a spinal cord injury, and just asked the question, does the CSF from those injured pigs make these neurons from the rat more active, sensitized? And the answer is yes. And it's fairly powerful. It's also fairly specific to the nociceptors, the pain receptors. That CSF doesn't really sensitize the sensory neurons that transduce like hair movement or vibration on your skin. So this was very interesting, and it tells us a little bit about what might be going on. Normally, the nociceptors have kind of a high threshold. They have to work at getting them activated, but after spinal cord injury, looks like they have a low threshold. So part of that might be an intrinsic property, part might be the extrinsic property. They might be sitting in a fluid that makes them more sensitized and more active. So we showed you that stretch requires nociceptors to induce a suppression of locomotion in the laboratory, and that might be reflected in some of the human condition. So how do those nociceptors do this? Looks like some of the CSF might have factors in it, which are causing those pain sensors to be more activated. Now, what about what those factors might be? A colleague of mine, Terry Walters, down at the University of Texas in Houston, did a great study. He's been one of the ones showing a lot about the activity of the nociceptors after injury. And he showed that macrophage migration inhibitory factor, MIF, is probably one of the key players in this, at least for rats. And the thing that happens there is that if they were to give MIF to spinal cord injury nociceptors, activates them at much lower doses than to sensory neurons that were not from a spinal cord injured rat. We did a nice set of experiments showing that MIF is probably involved here somewhere. So this is the work that we had done with the CSF shows the general concept. The Walters lab moved this to identifying a single candidate. Next, we'll need to figure out if that's actually reflected in human or if this is a rat-specific thing. Future biomarkers, right? So the CSF blood might provide us some biomarkers for these things. We're starting to identify specific ones out of those fluids. Biomarkers can be more than just molecular factors. They can also be functional. Sometimes it's things like walking speed. Some of the outcome measures can be biomarkers. Well, we're dealing with nociceptors here, and one of the questions, how do you know if you've activated a nociceptor? For somebody who is sensate or has a sensate part of their body, you can do pinprick, heat, pinch, noxious stimuli, and you just ask them, can you feel that? And they'll say yes or no. And if the answer is no, it doesn't feel painful, then you haven't activated the nociceptors, you haven't induced pain. But what about insensate areas of the body? We're talking here about below level parts of the body. So what happens there? How will I know if something is activating a nociceptor? This is a bit tricky. And there are no really good ways of doing that for insensate parts of the body. But we can learn some things from some other fields and some basic science. And interestingly, nociceptors, your sensory neurons that are pain receptors, at least the ones in the skin, have a really interesting property. Normally, they send their electrical signals from the skin where they're activated. It comes up through their axons and into the spinal cord where it communicates with spinal cord neurons. There's a really interesting thing that happens. Nociceptors have something called an axon reflex. And everyone listening to this has experienced the effects of an axon reflex, even if you don't know it. What happens there is when these axons are activated, and they would normally send their electrical signal, the other thing they do is release some neuropeptides and other communicator molecules into the tissue, and sometimes onto capillaries or onto arterioles that are sitting in the skin, in the tissue. And that release causes some things. If you get enough of that release, you get something called plasma extravasation. And what that does is it actually makes the blood vessels leaky. And you get fluid extravasation, and you get cells crossing from inside the blood vessels to outside the blood vessels, immune cells. And they do a little thing that looks a lot like this. They get to the wall of the vasculature, and they poke right through. Anybody who has a fenestrated garden hose, this is exactly what plasma extravasation looks like in the skin. This is what it looks like on the surface. When you get a mosquito bite, you get the red flare that goes around that, the wheal and flare. That is all due to neurogenic inflammation, plasma extravasation, etc. So if we want to ask, are nociceptors being activated in this particular situation with some folks who can't report to us? We may be able to do something akin to a mosquito bite, but not actually. But there are clinical tests where you can ask the question, what is the level of temperature that's required to induce this kind of response in the skin? Chances are pretty good that with spinal cord injured folks, that threshold is going to be lower than with spinal intact folks. And those sorts of things are done in other fields and other parts of neurology, looking for neuropathy, et cetera. We may be able to adopt these to get an idea about whether our spinal cord injured folks are having issues with their nociceptors. This is just thinking a little bit forward. So sensory input below a spinal cord injury may remain subliminal. Folks may not have the ability to perceive anything that's going on down there, but the effects can be real, even if they're not perceived. Effective intervention and trial design should not be agnostic to this. I think that it's not, but the question is, how are you going to deal with it? I think the development of biomarkers for sensory status, we really need to start identifying those, validating them. I wanted to thank a lot of the folks who were involved in this. I won't read all the names, but some of the funding agencies, colleagues, both at University of Louisville and outside. And I thank you very much.